Thanks very much for the opportunity to speak today. I'm going to give you a bit of a snapshot of what Surnova is about and talk about our clinical programs and our future. Please be aware of the forward-looking statements that I'll be saying throughout the presentation. So first of all, Sernova is a regenerative medicine cell therapy company, and we are focused on a combination of a medical device that is placed deep under the skin that provides an organ-like environment for the placement of immune-protected therapeutic cells. Our primary program is in type 1 diabetes, but we also have preclinical programs both in hemophilia A, and I think we're the first company in the world to be able to be working on benign thyroid disease, which I will talk a little bit about later. So some of our important highlights are that we have a phase 2 study ongoing that I'm going to be talking about and providing some really exciting data in type 1 diabetes. The second part is from the financial aspect. We have approximately $25 million in the bank, plus about $40 million in warrants that will be exercisable as our share price moves up, with a potential of approximately $70 million in the bank, which will get us through at least 2 years of our full development clinical programs. So what are our technologies about? So we have developed 3 parts of the technology, and the first is what we call the cell pouch device. And what we have found that is really important is that we need to have a device to be able to put the therapeutic cells in, and those cells need to live in a proper organ-like tissue environment. So when we developed the device, it is made up of a series of chambers that are very, very porous with single small plugs, cylindrical plugs in each chamber. And so what we do is we place this device deep under the skin at the level of the muscle around the belly, and it's about the size of a business card and a few millimeters thick. And over a period of about 2 to 3 weeks, the body will naturally fill in the chambers through the pores with highly vascularized tissue. So then we can go in after about 3 weeks, pull out the plugs, and then put the therapeutic cells in, and they're sitting in a natural tissue matrix. And this is very different than what other companies are doing, where they're placing cells into a device, putting them in the freezer, and then putting that device in the body where it fibroses. So what we have found is that by having the tissue incorporate into the device, it becomes a part of the body, and we eliminate that potential for fibrosis. So in terms of our therapeutic cells, because we're really trying to change the whole way that patients with type 1 diabetes are being treated, we wanted to start off with working with human donor islets to begin with. So our first clinical trial is with human donor islets, but we've also had a number of collaborations with major pharma companies that have advanced iPSC cells technologies, and we are working on bringing one of these technologies in and expect to have a worldwide exclusive license very, very soon. So in terms of our immune protection, our first clinical trial is using the latest anti-rejection medications, but we have also, after a number of years, found a technology that we call conformal coating, which is a thin cross-linked polymer coating that can coat the cells before they go into the device so that we can have an immune-protected macro device with the therapeutic cells. And our goal is to eliminate the need for any anti-rejection medications. So in terms of our approach, we believe we have the only biologically appropriate delivery system. And again, this is where we put the device into the body in a very short procedure and allow the vascularized tissue to grow into the device before we put the therapeutic cells in. Then we can go in and rapidly put the cells in, they engraft, and they start to function. So what work have we done over the past 10 years? So we've done significant number of studies around showing that the device becomes vascularized in small animals. We work with our large animal models. We use pigs specifically because the pig skin has a very similar architecture to human skin. We've also done non-human primate studies, and obviously we've done this in the clinic in humans. And we have patients that have had our devices in for over 3 years, and they're still functioning. So we're not getting that issue with fibrosis. We have worked with human donor cells. So in the islets, we've worked with tissues in the device, and we've been able to show that we're able to get those to function too long-term. And obviously, we've worked with stem cell-derived technologies. So in terms of our pipeline, we have 3 areas that we're working in. One is type 1 diabetes. The second one is hemophilia A, which I will talk about in a little while. And the third one is benign thyroid disease. And for each of those areas, we start off with working with the human donor cells, and then we move into stem cell-derived technologies. So I'm going to focus now a little bit on our type 1 diabetes program. And importantly, in terms of the clinical study that we're working on, we're working with patients that have severe hypoglycemia unawareness. So what is this? This is working with the patients that when they take an insulin injection, their sugar levels can drop to very dangerous levels and they can suddenly go into a coma and die. So these are the patients whereby the parents are having to stay and sleep with the children at night and wake them up and make sure that their sugar levels aren't dropping too low. And the only way you can help these patients is with a cell therapy approach, because it not only produces insulin, but it also produces the other hormones that can help regulate that blood sugar level if it drops too low. So in terms of our study design, we're working at the University of Chicago, and we have an investigator, his name is Dr. Piotr Witowski, that we've been working with over the past few years. In terms of our patient criteria, we're working with patients that have severe hypoglycemia unawareness and long-term diabetes, and the hypoglycemia awareness is occurring despite the best medical care. And in terms of our outcomes, we have a sentinel device that is very small that comes out after 90 days, and we look at the survival of the cells. So we'll show that too in just a few minutes. But we're also looking at reduction of hypoglycemia unawareness event, reduction of hemoglobin A1C, and we have approximately 20 other endpoints. So in terms of our study design, it's really quite unique because we're learning a lot about not only how the cells are working, but we're also learning a lot about dosing and how many cells are required to treat all patients. So what we do in our study is that we first implant the cell pouch. We wait approximately 3 weeks. We then stabilize the patients on the immunosuppression protocols, and once they're comfortable with that, then we can put the first dose of cells in. And very interestingly, what we have found over time is not just loading the device with as many cells as possible, but we actually found that a marginal dose of cells works much better. And so I can explain about why that is. And if you think about how islets work, they actually will read blood sugar levels in the local environment. So if you pack them all in together, they can turn each other off, just like having like 5 thermostats in a room. So the way the study is designed is that we can give a number of doses. We follow efficacy over a period of time, and then at the end of the treatment phase, we get the patient up to a certain amount of efficacy, and we're trying to get them to insulin independence. So we have the ability to put a small dose of cells in the portal vein, which is the standard procedure. And what we're doing right now is we're adding up all of the cells and making slightly larger devices, and I'm going to be placing that number of cells in the next set of patients. And importantly, we've been able to get patients to insulin independence. So our physician has presented data in case studies in a number of different conferences, including the American Diabetes Association. And you can see on the left-hand side at the bottom, this particular patient has a number of severe hypoglycemic events on a weekly basis. And what we were able to show with our islet transplant is that we eliminated the severe hypoglycemic events, and we also reduced the requirement for injectable insulin on a daily basis by approximately 50%. And then when we gave them the top-up, we were able to get that patient to complete insulin independence. So this is a little bit of a slide of what you see inside of our device. So we removed the sentinel device, and it might be a little bit difficult to see, but we're able to show significant survival of cells. We're able to show insulin. We show glucagon, somatostatin, which are the regulatory hormones, and we see C-peptide, which is a biomarker for production of insulin by cells. So it's really important to be able to show in your devices that these cells are actually surviving. We always talk about all of the biochemistry and this kind of thing, which is great for scientists, but what really matters is what the patients have to say. And after this patient became insulin independent, they wrote us a statement, and this person has been type 1 dependent for 47 years. They've taken over 20,000 injections, and this person has now been insulin independent for over 2 years. So this is what we call a functional cure and what we're trying to do for all of our patients going forward. The other thing that's really important is we now have 2 patients that are insulin independent. The first one is for 2 years, the second one is at 6 months. And importantly, if you look at this chart, you can see that every diabetic patient is really quite different from each other. And what we're trying to do is get the right dosing of cells so that we can treat all patients. And if you quickly look across the board here, you can see the hemoglobin A1C for the first patient is 6.5, but for the second is 9.2, which is really bad. But we've stabilized both of those patients back to normal with our cell therapy approach. And the same thing with the hypoglycemic events. The first patient had a large number of these on a weekly basis. Second patient wasn't so bad. We brought them both back down to 0. So the approach that we're trying to do is we want to treat the millions of patients with type 1 diabetes and get them with a cell therapy dose that can bring them all back to a normal state. So in terms of what we're doing as we're moving forward, the important thing that we have found is that the product is very, very safe. We actually have our first patient that was asked by our clinician if they would like their devices taken out after 2 years, and they refused because they're doing really well. So the safety of the devices and the cells is very, very positive. And from an efficacy perspective, our most advanced patients are continuing to do well, and the other patients in the study are moving through the various processes of the cell therapy. So where are we going next? So we're going to be releasing additional data as we're going forward in 2022. We are aggressively pursuing our next iPSC stem cell-derived technology, so there'll be a lot more exciting information to hear about that in the near term. We are advancing our conformal coating technology. We have a significant collaboration with the University of Miami right now, and the idea there is that we'll be bringing this conformal coating technology into the clinic as quickly as we possibly can. The other thing that's really important is, are these treatments going to be paid for? For type 1 diabetes, the cost of type 1 diabetes is not insulin. It's really the side effects of the disease. So we conducted a payer study with physicians and insurance companies, and they have come back, and with our therapy that we're working on, they have said that they would pay between $100,000 and $200,000 per treatment per patient. So this is, so the real cost of the issue here with type 1 diabetes is really the side effects of the disease, and insurance companies do not have a problem with paying this kind of fee. How it's going to get paid will be determined as things are going along. So conformal coating technology, again, it's a cross-linked polymer technology that's been developed over a 12-year period of time. And the really exciting thing, the important thing about this is that the kinetics of insulin and glucose release are the same with the coating as without the coating, and that's a critical part of this. And so we're now just moving through the studies with Dr. Tomei at the University of Miami to get this into clinic. So with respect to our thyroid program, we're working with patients, and it's about 150,000 patients in North America alone who are having their thyroid gland removed. And if you think about the thyroid gland, it's really a lot like the engine in your car. So if you remove it, your energy level drops significantly. There's significant obesity that occurs and depression. So what we're looking at is being able to take the healthy tissue that's remaining from the thyroid gland, and we've been able to put this back into cell pouch in preclinical studies and shown long-term survival of the cells, and also bringing back the feedback loop of the thyroid hormone. So we're going to be moving this program into clinic by the end of this year, early 2023. Our other program is hemophilia A, and here we had a grant with the Horizon 2020 group with a team of academic investigators in Europe, and we were able to develop an ex vivo gene therapy product where we take a sample of the patient's blood, isolate the endothelial cells, we inserted the gene for factor VIII using a lentiviral approach, and then we were able to scale up those cells and place them in the device. And in preclinical models, we were able to return the function of clotting, and we're going to be moving this program into clinic also. So this is a great example of an ex vivo gene therapy product where the cells are placed into a device in a very, very safe environment. So we're looking forward to bringing this on, back home also. So in terms of our, more on the share side, Sernova is traded on the Toronto Venture Exchange. We're transitioning right now to the Toronto Big Board, and we have aspirations for NASDAQ in approximately a year. We are also on the Frankfurt Xeter exchange, and we have a market cap of approximately $400 million at this point in time in a strong cash position. So thank you very much, and really appreciate the opportunity to speak today.